Hello, my dear students. Welcome back to Physio-Pulse channels. In today's lectures, we will discuss about the concentration of the urine and in a simple conceptual exam-oriented manner and specially designed for undergraduate students. So let's get started our concentration of urine. And in these chapters, we will segregate in the two lectures, especially in first lectures, we will discuss about the introductions and second medullary gradient and the countercurrent mechanism that is countercurrent multiplier as well as the countercurrent exchanger. But we will discuss in the first lecture countercurrent multiplier. But in our consecutively second lectures, we will discuss about countercurrent exchangers, role of ADH, and summary of urine concentration. And after that, we will discuss some applied physiology. So let's start our introductions. Every day, we know that 1 liter of glomerular filtrate is formed with the large quantity of water. If this much of water is excreted in the urine, body will face a serious threat. So the concentration of urine is very essential. So therefore, osmolarity of the glomerular filtrate is the same as the plasma, that is the 300 milliosmol per liter. But normally urine is concentrated and it is there is a 4 times from the normal level, that is the 1200 milliosmol per liter. So therefore, osmolarity of urine depends upon the two factors. Number one, water content in the body. Number two, antidiuretic or ADH hormones. So now mechanism of urine formation is the same of the dilute or urine and the concentrated urine till the fluid reaches the distal concentrated fluid. However, the dilution or concentration of the urine depends on the water content in the body. Now first, formation of dilute urine, how it is formed. Actually, when the water content in the body increases, the kidney expels dilute urine. That means now this is achieved by the inhibition of the ADH secretion, because you know that ADH has a role for the reabsorption of water as well as the sodium and chloride molecules. So it's leading to excretion of large amount of water, and this makes urine can dilute. Now, this dilution process is involved. That is the dilution and even also concentration process. They're also just opposite into the dilute urine formations. That is what happens now when the water content in the body increases. That means kidney retain the water and excess concentration of the urine. Formations of the urine, concentrated urine, is not as simple as that of dilute urine. So therefore, it takes the two processes. One is the development and maintenance of medullary gradient by countercurrent system and secretion of ADH. So these two specific topics we will discuss here. Number one, how medullary gradient can make urine concentrations through the countercurrent systems. So to understand of this thing, medullary hyperosmolarity, actually this term you have to understand. Particle interstitial fluid is isotonic to the plasma with the osmolarity of 300 milliosmol per liter, and this osmolarity near the cortex is also same. However, while proceeding this outer part of the towards the medulla, then the osmolarity increases gradually and it reaches the maximum innermost part of the medulla near the renal sinuses. And here the interstitial fluid is once again it makes the hypertonic and it is 1200 milliosmoles. So this type of gradual increases osmolarity or medullary interstitial fluid is called the medullary gradient. So you can understand the medullary gradient, what is the definition of medullary gradient, and it has important role for making the concentration of the urine. And development of maintenance of medullary gradient. Therefore, a kidney has some unique mechanism, countercurrent system, which is responsible for development and maintenance of the medullary gradient for hyperosmolarity or of the interstitial fluid in the inner medulla. So how it is happened? If you see the countercurrent system, what happened? The countercurrent has a mechanism. There is a system where the U-shaped tubes is there, in which a flow of the fluid is opposite direction in the two limbs. That is the U-shaped tubes. So there are two divisions. One is multiplier, which is formed by the loop of Henle, and countercurrent exchanger, which is formed by the Vasa Recta. Now, multiplier, it has loops of Henle. Loops of Henle functions as a countercurrent multiplier, and it is responsible for development and hyperosmolarity of the medullary interstitial fluid and medullary gradient. Now, the role of loop of Henle in development of medullary gradient. Loop of Henle of juxtamedullary nephrons plays a major role as countercurrent multiplier because loop of these nephrons is long and extends up to the deeper parts of the medulla. Main reason for the hyperosmolarity of the medullary interstitial fluid is the active reabsorption of sodium ion, or sodium chloride, and other solids from ascending limb of loop of Henle. into the medullary interstitium. These solutes accumulate in the medullary interstitium and increase the osmolarity. Now, due to the concentration gradient, the sodium and chlorine or chloride ions decreases from the medullary interstitium into the descending limb of loop of Henle and reaches into the ascending limb of loop of Henle via the hairpin nodes. So you see, if you see this 300 milliosmol gradually increases to 600 milliosmol due to the sodium ion and chloride ion reabsorptions, and ultimately whenever it reaches into the loop of Henle, it gradually it increases the medullary intestine. The value is osmotic pressure is or osmolarity is 1200 milliosmoles. Now, these portions, this 1200 milliosmol, it will, whenever it reaches to the thick ascending loop of Henle, descending loop of Henle, sorry, ascending loop of Henle, at that time, it gradually decreases. So, thus the sodium and chlorine ion repeatedly recirculated between the descending limb and ascending limb of loop of Henle, and making a urine is concentrated here. So apart from this, there is a regular addition of more and more new sodium and chloride ion into the descending limb of Henle, and thus the reabsorption of sodium chloride from the ascending limb and adding the new sodium chloride ions to filtrate or increase the osmolarity pressure, osmolarity of interstitial fluid or medullary gradient. So it is called the multiplication or multiplier. So we students, you clearly understand that why it is called the counter loop of Henle is called the countercurrent multiplier. Some other factors responsibility for making the hyperosmolarity, like the sodium reabsorption sodium ion from collecting tubule, like recirculation of the urea, and even also this number one, number two, these two important point that can make the medullary part of the collecting duct into the medullary interstitium, that the reactions or osmotic pressures are gradually increases. Now, if you see the countercurrent exchanger mechanism, then it will be clear that the Vasa Recta, that is another part of the nephron or urinary structures, it can responsible for the exchanging of the exchanging the ions, that is called the countercurrent exchanger, and which is for maintenance of the medullary gradient and development of the countercurrent multiplications. So how countercurrent multiplication or role of Vasa Recta in maintenance of medullary gradient, that we will discuss in our next lectures. So I think students, you all have understand what is the countercurrent mechanism, how urine is concentrated by the specifically two important point, medullary gradient and that countercurrent mechanism, that is the countercurrent multiplier we have discussed. And in our next lecture we will discuss countercurrent exchanger and even also some important applied physiology and role of ADH, specifically antidiuretic hormone, how it creates into the distal converter tubule. So students, thank you for watching this lecture. I hope this session has helped you understand the topic clearly. If you found this video useful, please like, share and subscribe to our Physio-Pulse channels. Do not forget to press the notification bell so you never miss our upcoming lectures on physiology, biochemistry or biology. If you have any queries, any question or any suggestion, please write some in your comment box. And thank you for watching this once again. I will see you in the next lectures, and until then, keep learning. Thank you.